Hi everybody, welcome back. Before we start talking about DNA replication, let's get an update on the coronavirus outbreak. We are currently just over 73,000 confirmed cases. Again, most of them in mainland China. Now the number outside of China has gone up a bit because of all the cases on this Diamond Princess cruise ship. You put a lot of people together in a closed environment, introduce an infected person, and you have an outbreak on the ship. We will return to cruise ships later when we talk about gastroenteritis. We also have 1,875 deaths, and remember, the deaths are likely to go up. It's probably underreporting. Now, an interesting addition to the Johns Hopkins page here is that now on this graph of cases, they have a daily increase. So let's take a look at that. First, just the total number of cases. And you'll see that on February 13th, there was a huge jump, and that's because they changed the method of diagnosis. They felt that PCR was missing a lot of positives. They started doing MRI to look for lung infections. And according to this, it looks like it's tapering off. Let's look now at daily increase. And here is a wonderful curve where you can see the number of cases each day, not cumulative. And here on February 13th and 14th, those big spikes. And then it's gone down. In the last 3 days, the 15th, the 16th, and the 17th, it looks like it's leveled off. I mean, there are 2,000 a day. It's not insignificant, but the number of cases have leveled off. And then today, as I record, it's in the morning, and we probably don't have complete reporting yet. But that's a nice addition to the website. I think the daily case number really gives you an idea of how the epidemic is going. Today I want to talk about viral DNA replication. Last time we talked about how viruses with DNA genomes are transcribed. Today we'll talk about how the DNA itself is copied to make more genomes, which of course is needed to make new virus progeny in an infected cell. And here are the players for today. Class 2, the Parvoviruses with single-stranded DNA. We won't talk about hepatitis B virus with its gapped DNA, but it has to be repaired to make a double-stranded genome, which is then replicated by some of the mechanisms that are common to the other viruses we'll talk about today. We'll spend most of our time on the group 1 viruses, adenoviruses, Herpes simplex virus, polyoma and papillomaviruses with double-stranded DNAs, and we'll also throw in a bit on Poxviruses, which aren't listed here. And remember, as we said last time, viral DNA replication is always delayed for the reason that at least one and sometimes more viral proteins must be synthesized in order to start DNA replication. And so that would include repair of genomes, transport in the nucleus, which would happen first, transcription, export of mRNA, synthesis of the protein, often going back in the nucleus, so it takes time. So DNA replication is always delayed, and that's why, as we saw last time, the synthesis of capsid proteins are delayed, because there's no point in making those until you have DNA in which you can use to insert into them. DNA replication has universal rules, much like RNA replication. The DNA is synthesized using a template to direct the incorporation of dNMPs. And of course, the template is read in a 3' to 5' direction. The new chain is synthesized in a 5' to 3' direction. And DNA replication always takes place via what we call semi-conservative replication. There are 2 daughter strands. So here in the middle of this image is a DNA, a double-stranded DNA, that is going to be replicated. Each strand is copied to make double-stranded progeny. That is semi-conservative replication. Conservative, it would be only copying one strand. So in this case, the green strand is copied twice to make progeny. So that's semi-conservative replication. That's what happens with these viruses that we're going to talk about today. DNA replication initiates at specific sites on the template, which we call origins of replication, or just origins. Of course, the actual synthesis of DNA is catalyzed by a DNA-dependent DNA polymerase, but we always need accessory proteins. And DNA synthesis may be primer-dependent or primer-independent. In every year of this course, up until this year, I have always said DNA synthesis is always primer-dependent. But just in the past year, some new polymerases were discovered that carry out primer-independent synthesis. And so our dogma is overturned. We now have primer-independent DNA polymerases. And so here's an experiment that demonstrates this. And what we're looking at is a polymerase reaction with DNA polymerase, template, radioactive dNTP, and we're measuring incorporation of TMTP on the y-axis. And so the standard polymerase here, T7 DNA polymerase in red, you can see you make nice DNA product with time, which of course is the x-axis. If you leave out the primer from the DNA polymerase reaction, you don't make a product. Primer-dependent DNA polymerase. However, we have a different polymerase on this slide. These are purified proteins, by the way. It's called bacteriophage NRS1. This is a bacteriophage that infects bacteria that inhabit those deep sea vents at the bottom of the oceans. Really, really deep down, high pressure, hot, really amazing environment. There are bacteria that grow there, and there are phages that infect them. So this one, NRS1, its DNA polymerase works well with or without a primer. And blue with a primer, without a primer, and blue and green with a primer. You can see there's no difference. Primer-independent DNA polymerase. And there's one other example of this that's been found as well. And these are ancient polymerases, which suggests that primer dependence evolved later. The mechanism of DNA synthesis is very similar to that of RNA synthesis by DNA-dependent RNA polymerase or RNA-dependent RNA polymerase. Here on the bottom is a RDRP of poliovirus. Remember we said it's a right hand with the active site in the palm domain and the thumb and fingers, in this case, touching to make a closed polymerase. Here's the structure on top in red of a DNA polymerase. And here's the active site in the yellow beta strands. And that's a DNA coming through being synthesized. And you can see the fingers and the thumb domains are not touching in this DNA polymerase, but the overall architecture is quite similar. The key here, of course, is that the active site is where DNA synthesis occurs. And DNA synthesis occurs by similar mechanisms for RNA synthesis. Shown on the right, this is a picture we saw before when we talked about RDRP. We have a template lined up on the right from 3 to 5 prime with 4 bases, sugars, and phosphates in between. And then we have a growing product. In this case, we have a primer and then 2 bases. And here the third base, a T, is being added, matching the A template. The T comes in, of course, as a triphosphate. 1, 2, 3 phosphates. And then there is a 2-metal mechanism of catalysis, 2 magnesiums here, held in place by our aspartate residues, just like we saw for the RNA polymerases. And they catalyze nucleophilic attacks to liberate the pyrophosphate, the 2 Ps, and you end up with just the alpha phosphate left between the 2 bases. So the mechanism very similar between DNA and RNA polymerases. And again, that together with the protein homology among these enzymes shows that they all evolved from a common ancestor. Now, viruses require assistance from the host. Viral DNA replication always requires the synthesis of at least one viral protein, and sometimes many, depending on the size of the genome. And that's, again, why DNA replication is always delayed after infection, because you have to make this protein and it needs to start DNA replication. Those viruses with small genomes, they need more host proteins. They need the DNA polymerase and many other proteins. The bigger viruses, as you will see, they encode many proteins required for DNA replication. Complex viruses like Poxviruses encode all the proteins needed for DNA synthesis. So they are distinct from, say, the polyoma and the adenoviruses. The polymerase, again, for small DNA viruses, comes from the host. The small viruses, their genomes are not big enough to encode a polymerase. They typically encode a protein or proteins that, what we say, orchestrate the host, and you will see they trick the host into replicating viral DNA genomes. And this includes the polyomaviruses, the papillomaviruses, and the parvoviruses, which we'll talk about today. They are replicated by the host DNA polymerase, but they all encode a protein to make sure that the host polymerase recognizes the viral DNA. The larger DNA viruses encode most of their own replication systems, including herpesviruses, adenoviruses, and poxviruses. So the viral proteins that are involved in DNA replication, again, the smallest ones don't have many of these or any of these, but the bigger ones do. A DNA polymerase and accessory proteins, typically an origin binding protein, helicases to unwind templates, exonucleases, and enzymes of nucleic acid metabolism like thymidine kinase, ribonucleotide reductase, and DUTPase. So viral genomes don't encode all of the enzymes of nucleic acid metabolism, but they encode some, presumably to make more metabolites that they need to make lots and lots of genomes. Our first question is, which statement about viral DNA synthesis is not correct? A, large DNA viruses encode many proteins involved in DNA synthesis. B, small DNA viruses encode at least one protein involved in DNA synthesis. C, viral DNA replication is usually delayed after infection because it requires the synthesis of at least one viral protein. And D, all DNA polymerases are primer dependent. The answer is D. As I showed you, there are now some DNA polymerases which are primer independent. Every other statement is true. Large DNA viruses encode many proteins involved in DNA synthesis. Small DNA viruses encode at least one protein involved in DNA synthesis. And viral DNA replication is usually delayed after infection because it requires the synthesis of at least one viral protein. DNA viruses have DNA genomes with diverse structures, which we already know. We have seen these before. For example, the single-stranded DNA genomes of adenovirus associated virus, which is a parvovirus. By the way, parvovirus is the family, and within it are adenovirus associated viruses, often used for gene therapy, and the viruses that cause disease of humans and pets. These have unusual terminal repeats, and we will talk about how this is replicated. Of course, SV40 polyomaviruses and the related papillomaviruses have a double-stranded circular DNA genome. Human adenoviruses, a double-stranded linear DNA genome. Same with herpes simplex virus, only longer. And finally, vaccinia a poxvirus, a very long DNA genome with a double-stranded DNA that is covalently linked at the ends. There are no 5' and 3' ends that are free. They're joined with one another. We call that a terminal loop. If you denatured this DNA, it would be a single-stranded circle. Let's talk about how these DNAs replicate. First, an overview of the two mechanisms of double-stranded DNA synthesis that we're going to talk about today. On the left is what we call a replication fork. Here, the DNA, the double-stranded DNA, is being copied on both strands. And RNA primers, shown in green, are used to prime DNA synthesis. So obviously this requires the double-stranded DNA to be denatured, so you have two single strands. And synthesis is occurring on both strands. On one strand, it's continuous because it can go in a 5 to 3' direction. But on the other strand, it is discontinuous. You have to put down a primer, synthesize a piece of DNA, another primer, synthesize a piece of DNA. And the reason you have to do that is because this first primer here is exposed as the fork grows, as the DNA is denatured, and so that is made, its product is made, but the upstream product isn't available to be made until the replication fork proceeds more. So you have to add little pieces as this fork opens up. So that's a replication fork. It is primed by RNA primers, and it can be found in the genomes of papilloma and polyomaviruses, herpesviruses, and retroviral proviruses. The provirus is the retroviral genome integrated into our double-stranded genome. It replicates along with our genome, and that happens by a replication fork using RNA primers. The other mechanism is called strand displacement. And here we start with the double-stranded DNA, and we only copy one strand at a time. Eventually the other one will be copied, so this is semi-conservative replication. But first, the first strand is copied, and that bumps off the second strand, and we'll see later the second strand is then copied. This first strand synthesis is never RNA primed among the adenoviruses, parvoviruses, and poxviruses. The primer, and these viruses are all requiring a primer, the ones we talk about today, varies according to the virus. For adenoviruses, it's a protein, and for parvo and poxviruses, it's a DNA primer, which is part of a hairpin, and you'll see how that looks like in a moment. So strand displacement is distinct mechanistically and also by the primer that is used. When we replicate DNA, we have what is called a 5' end problem. And let's see what that is. So on the top we have a DNA template, a linear DNA template. We are putting down RNA primers. And if it's discontinuous, we put down many. If it's continuous, we can put down one and extend it. But here we're using several as an example. So these RNA primers are elongated to make DNA, which is shown in red. Eventually the primers are removed. The gaps are filled in by polymerase. Then the DNAs are ligated or joined chemically together so that they're one continuous strand. And we end up what's shown on the bottom. And you can see we're missing a little piece at the very left end of this DNA where we removed that last primer. The rest of the strand is fine. But on the left, if this is the end of the DNA, we are left with a gap. And that is the 5' end problem. And there are a variety of ways to solve it. And we will see how this problem is solved during replication of DNA viral genomes. Let's start with SV40 DNA replication. This was a virus discovered in the 1950s and which drove much of our early understanding about DNA replication. It was very difficult to study the synthesis of DNA in cells because the DNA is huge and it's difficult to purify. However, SV40 DNA is about 5 kilobases of double-stranded circular DNA. It comes packaged in a virus particle that you can easily purify. You can extract the DNA and add it to extracts of cells to study DNA replication. And so we know so much about DNA replication from studying SV40. That's why I call this Lessons from SV40. And the first thing we know, which we now know to be common, but for SV40, we found that DNA begins at a very specific place on the template. It doesn't begin just anywhere. It begins at an origin of replication, which is shown here on the double-stranded DNA. If this were a clock, this would be 12 o'clock. And that's where DNA synthesis begins. It also turns out that that is where the 2 promoters are for early and late transcription. So it's all in one place, the 2 promoters and the origin, as you'll see in a moment. The way this DNA is replicated is via bidirectional replication from this one origin. So starting on the left here in panel B from the origin, the replication of DNA occurs on both strands. It starts at the origin and proceeds in both directions. So both strands are copied, and replication is bidirectional. And of course, this is a replication fork, which is one of those mechanisms of DNA synthesis we talked about. And that, of course, is primed with RNA primers. The reason we know that this starts at an origin and proceeds bidirectionally is from an experiment like the one shown in panel A, where the SV40 genome was cut once with a restriction enzyme to linearize it and then incubated in an extract from cells to watch DNA being replicated. And you can see the first panel, there's a tiny bubble at the left end because the enzyme used to linearize the genome was not at an equal place from both ends. And it's growing, and it's getting bigger and bigger. And you can tell the distance from the ends that it's growing bidirectionally. So that bubble there, we call it a bubble, is a replication fork, bidirectional replication from a single origin. Here's a schematic of what's going on. And not only is it bidirectional, but it's what we call semi-discontinuous. Here's the origin of SV40 replication. The polymerase or the DNA synthesis has proceeded some way away from the origin on both strands. And we say semi-discontinuous because there are both leading and lagging strands. So what does that mean? Well, when you first start DNA replication at the origin, the double strand denatures a little bit, not as much as shown here, just a little bit. And a RNA primer is added, and then you can start synthesizing DNA, shown in red. That's the leading strand. As the double-stranded template denatures more and more and the bubble gets bigger, this leading strand can get longer and longer. It's continuous. The leading strand is continuous DNA synthesis. There's also a leading strand on the other DNA strand, shown at the bottom right here. However, there are also lagging strands, and that is also what we call discontinuous DNA synthesis, because you can't synthesize in a 5' to 3' direction the same way as the leading strand, of course. You would have to, it would be backwards. So you put down, as soon as the DNA is denatured enough, you put down an RNA primer and you get a short piece of DNA made, and as the DNA gets denatured more, you can put down another primer and another one. So you have these short pieces of DNA that are made on both strands again. That's the lagging strand synthesis, and eventually they're all ligated together. And that's why we call it semi-discontinuous, because there is some continuous and some discontinuous synthesis. Now, the beauty of this mechanism, this is a circle. So there's no end problem. If this were the end of the circle, say, this leading strand could go right up to this discontinuous strand. The primer could be removed, filled in. You wouldn't have an end. So there's no end problem because there's no end on a circle. Very simple. So the end problem has been solved for viruses with circular DNA genomes because the circular configuration doesn't lead to an end problem at all. Here's the origin of SV40 replication. At the top, it's a wide view. Here's base 1. And the early promoter is shown here with an enhancer. And if you look in this blue area to the right of the early promoter, you can see the actual origin. And we'll look at origins in more detail later, but this one you can see has a number of different elements. It has a T-rich element. It has a palindrome, which is labeled LT binding site, and that's large T antigen or large T protein. And you can see there are 2 sets of binding sites for LT binding site 1 and 2. It's typical of an origin of replication. It's often mixed up with the promoter region with these viruses. So how does this all work? The viral DNA gets in the nucleus, and there, for SV40, remember, you have early transcription, which gives rise to the production of large T antigen. And there's no DNA replication until large T is made. Large T comes in the nucleus, and it binds the viral DNA at the origin. And you can see here, there are hexamers of large T binding on either side of the origin. This is labeled minimal origin because you could cut this out and put it in another DNA and it would get replicated. So the 6 copies of large T bind at either side of the origin. But this binding does a couple of things. It starts to denature the double-stranded DNA, and it recruits a cell protein called RPA. RPA binds large T. RPA coats single strands as large T unwinds the DNA. So T has 3 to 5 prime helicase activity. As large T denatures the double-stranded DNA, it also binds RPA, and then RPA binds the single strands to keep them single-stranded for the subsequent steps of DNA synthesis. So that's how large T recruits the cellular DNA synthesis apparatus to its genome. Remember, there's probably one or a few SV40 DNAs coming into the nucleus, and they are going to be ignored because the cellular DNA far outnumbers it. But this protein, large T, manages to grab RPA and start DNA synthesis. So that's what we mean when we say these little viruses need at least one protein to orchestrate DNA replication. All right, so now we have a little bit of a bubble here, single-stranded DNA on both sides. What happens next? The next thing that happens is that an enzyme comes in via binding to RPA and large T again. So we're doing more recruiting of the DNA synthesis apparatus. So Pol alpha primase is recruited by binding to RPA and large T. And this enzyme synthesizes the RNA primers and then begins to make short DNA fragments. And there's going to be more than one enzyme, DNA polymerase, involved in this, as you'll see. So here is Pol alpha primase making RNA primers on both strands. Next, we have a couple of other proteins that come in, RFC and pCNA, and these help to assemble what's called a sliding clamp that moves along the DNA, and eventually will bring the polymerase with it. And finally, polymerase epsilon, the purple oval there, comes in and starts to make long DNA. So the Pol alpha kind of gets things going, makes RNA primers, starts to make a little bit of DNA, and then the heavy work is done by polymerase epsilon. And here we're making, on both strands again, a double-stranded copy. And so that is shown here in more extensive view. Again, we have our polymerase epsilon in there. Another polymerase delta comes in to assist with DNA polymerization. And now you can see we're making leading strands, and on both DNA template, there's a leading strand, of course. Once this bubble is big enough, you can start making lagging strands. I mean, again, the origin defines where the leading and lagging strands start. And of course, let's look at this. Here's a leading strand going from right to left on the top and from left to right on the bottom. And then the lagging strand would go from right to left on the top, but in short bits, because only when the DNA template is exposed. And eventually this bubble gets bigger and bigger. The RNA primers are removed by RNase H, which takes out the RNA only. The gaps are filled in. DNA ligase makes a complete molecule. And here's a picture of the DNA replication machine of SV40. Again, we have a double-stranded template. 6 molecules of large T have bound to it, and this will be moving upwards, denaturing the DNA. Meanwhile, we have really 2 DNA synthesis machines. We have one here for leading and one for lagging strands. So both strands are replicated. And here you have polymerase epsilon and the primase beginning to make an RNA primer here on this strand on the right, and the sliding clamp that contains polymerase delta. So polymerase epsilon and delta are doing most of the synthesis here. Now, as you make DNA on a closed circular double-stranded DNA, it becomes twisted. And that's where topoisomerases come into play. So here's our SV40 genome, covalently closed circular DNA. Comes in the cell, it's replicated, again, bidirectional replication from an origin. So here part of it gets unwound. As you unwind part of the double strand, the rest of the DNA gets overwound because that unwinding is transferred to the rest of the molecule. And eventually this overwinding would stop DNA synthesis because the DNA would be all tightened up like a rope, all twisted. So what happens during replication, enzymes in cells called topoisomerases, and there are two of them, one or two, they cleave one strand of the DNA and unwinds it, and it causes what we call the formation of relaxed supercoils, cleaves it and religates it. And this happens periodically during replication so that you can get synthesis of the entire DNA. When you're done, you have two molecules made, the parental and the daughter strand, and it's, again, semi-conservative, so both strands are copied. But the problem here is that they are linked, be like two circles intertwined. If you take your thumb and forefinger of both hands and put them together, you get an idea of what's happening. Those two strands can't separate, and that's where topo 2 comes into play. It cleaves both strands so that it can separate the two products. And this is what we call terminal resolution. That's a term I'm going to use subsequently as well, when you separate the two products so they can go off. And of course, they're replicated again, over and over again. Our next question, the SV40 genome is a circular double-stranded DNA. Which statement about its replication is correct? A, the viral T antigen binds and unwinds the ORI. B, replication is bidirectional from a single ORI. C, the 5' end problem is solved. D, has leading and lagging strand synthesis. And E, all of the above. Which is correct? The answer is all of the above. The T antigen binds and unwinds the ORI. Replication is bidirectional from a single ORI. No 5' end problem because there are no ends. It's a circle. And we have leading and lagging strand synthesis, so it's all correct. Let's take a look at another kind of viral DNA genome, that of parvoviruses. Adenovirus associated virus. These are single-stranded DNA genomes with unusual terminal repeats that form these T structures or hairpin structures. There's one at each end. These are relatively simple genomes. They encode not too many proteins. A capsid protein, remember these are T equals 1 capsids. They're made up of one protein, so that's the capsid, or for cap. And rep encodes proteins needed for DNA synthesis. And the reason these ends are in this T shape is shown at the bottom. This is the nucleotide sequence of the very ends of the DNA. Starting at the 3' end, there's a hydroxyl, of course. And you can see that is base paired with the remainder of the DNA, and even the end is base paired to form this little T structure. Why this happens, you'll see in a moment. But of course, 3' hydroxyl is very important because it's where DNA synthesis is going to begin, and that's the primer. So these viruses replicate their genomes with a DNA primer that's built into the genome, and that's the origin of replication. That's where DNA synthesis begins. That's where the replication machinery binds. So that is the origin of replication. Here's how replication occurs with these viruses. So we start with the viral genome, which is in the particle. The particle, of course, enters the nucleus and the genome gets out. And the genome is shown here with the terminal hairpins. And at the top, we've extended it, denatured it, so you can see how these terminal hairpins form. And so A would be complementary to A', and that's how you can get these A' structures, and on the other end, A and A'. So A and A' form those base paired regions, and then the ends are complementary as well, but it's not reflected in the labeling. All right, so this DNA gets in the nucleus. Remember, it has to be repaired first, because the first thing that has to happen is transcription to make a protein needed for DNA replication. So a cell polymerase, delta, actually fills in this gap, and it goes all the way through the right end, and you get this molecule shown here after step 2. So now we have the left terminal hairpin. The 3' hydroxyl has been the site of synthesis by polymerase delta. The red is the new strand made, and it copies all the way through the right hairpin. So the polymerase can melt through that. And then this DNA is transcribed to make an mRNA encoding this protein Rep7868. And Rep7868 has a very important function, and that is it binds the origin on the left end, but it also makes a nick between A' and D where that arrow is. It's a sequence-specific nick. It's always at the same place in the DNA. Its function is to make a 3' end so you can get DNA synthesis. And the 3' end is shown there at D. The cellular polymerase then elongates it, and it's recruited in part because this Rep7868 protein is like a T antigen in a way. It's recruiting the polymerase to it. So now you have a double-stranded copy of the genome, a replication intermediate. The ends make hairpins, and then the left end makes hairpins, and then you can start initiation again at the 3' hydroxyl of the red hairpin, as shown there. And this synthesis is strand displacement. So the DNA is copied, and it's displacing the top strand. You eventually have the production, the displacement of the top strand, which is shown here. So the left end is not forming a T, but the right end is forming a T. Meanwhile, that synthesis is completed. It's displaced this strand. So now on the bottom here is the DNA whose synthesis has been completed. So that's one strand from the incoming DNA and one copied strand, and that will go through this entire sequence again. So it will be nicked and elongated, and then followed by strand displacement. And eventually both strands will be copied that way. And this strand right here can be copied as well by the same mechanism, so both strands are copied. So you can see we have continuous replication. There's no need for a lagging strand here, because it's all strand displacement from the ends. The inverted terminal repeat is the primer, so no polymerase alpha is required, which is the primase, as we saw before for SV40. And the polymerase delta is the one that copies this genome. The Rep6878 are needed for initiation. It cleaves at this fork, and resolution also. So in this case, there's no replication fork, but DNA synthesis is by strand displacement using a DNA primer, which is built into the genome. Let's talk about adenovirus, larger viruses with double-stranded DNA genomes. In this case, there is an origin of replication at both ends of the DNA, as you can see labeled by ORI, and this occurs by strand displacement synthesis. Now you can see here at each end of the DNA, at the 5' end, there's a protein called TP. That stands for terminal protein. It's also in the packaged virus DNA in the particle. And that's the primer for DNA synthesis, as you will see. The way this begins is at one end of the DNA. We have a polymerase molecule. This is a virus-encoded DNA polymerase in purple there. And it is non-covalently bound to a small protein, also a viral protein called pre-terminal protein. The polymerase adds a single C residue to PTP, which base pairs with the viral DNA. You then have priming of the first 3 bases here. It slips back so that it's even with the 3' end. And then at this point, the polymerase detaches from PTP and begins to copy the genome, and PTP is left bound to the first C. So that PTP-C is the primer for DNA synthesis. And DNA synthesis occurs by strand displacement. So this whole process on the left here is an enlargement of what's happening here. This is occurring at one end of the genome. The polymerase is then displacing the top strand and synthesizing a copy of the bottom strand in red there. And as the top strand is displaced, it is bound by a virus-encoded DNA binding protein, this yellow sphere, to keep it single-stranded. And so at the end, you have, once the polymerase traverses the entire genome, you now have a double-stranded DNA of an original light blue strand and a new red strand. And that can go through this sequence of events again. The pre-terminal protein and the polymerase starting an initiation. As I said, the polymerase links the C to this protein. It links it to a serine residue, but it only happens when the polymerase and the PTP are ready to do an initiation at the origin of replication. Last thing I want to tell you about before we move on to the bottom part here is this pre-terminal protein is cleaved by a viral protease to become the terminal protein, which is then part of the viral DNA in the packaged particle. So this is the synthesis here in the upper part of one strand. The displaced strand is shown here, the displaced strand, which is eventually all coated in DNA binding protein. Its ends are inverted terminal repeats. The ends of both ends of the genome are inverted terminal repeats, which was shown here in this picture, ITR, ITR, and those can form base pairs, shown here, A and A prime, and that looks to the polymerase like the end of the genome. So the polymerase latches onto it and begins copying it by the same mechanism as shown here. It is now making a light red strand on the bottom here, displacing the DNA binding protein, and eventually it is double-stranded. So both strands are replicated by this mechanism, as you can see. And every time you do this displacement synthesis, then the bottom strand, there's a little bit of displacement of the double-stranded portion, but then it's simply extension. There's nothing to be displaced as there is here. So again, there's no end problem because the primer is protein linked and it begins essentially at the very end of the genome. So you don't have to have an RNA primer and you don't have to worry about a gap. The DNA binding protein of adenovirus, it's a viral protein which coats the single strands. And here's a model of the parental DNA being copied by the polymerase. This is strand displacement. As I said, as the top strand is moved off, it's bound by the DNA binding protein. We know the crystal structure at high resolution of this protein. It's shown on the left here, and it has a very interesting shape. It has a globular body, as you can see here, which is depicted in the drawing, but then there's an extension of the protein. We think this helps fit into the double strand, help denature it. So as these proteins are binding, this little extension is helping to denature the double strand to allow the polymerase to synthesize the complement. Our next question is, how is DNA replication of Parvovirus and Adenovirus similar? A, they both require protein-linked primers. B, replication occurs by strand displacement. C, DNA synthesis occurs in the cytoplasm. D, a replication fork occurs in both. E, none of the above. How are they similar? The answer is replication occurs by strand displacement. They don't both require protein-linked primers. That's only adenovirus. Parvo, of course, uses a DNA primer. This does not occur in the cytoplasm. It's occurring in the nucleus. And there is not a replication fork in both. They are both strand displacement. Another virus with an even larger genome and hence encoding more proteins, herpes simplex virus. Envelope viruses with a nucleocapsid containing a very long double-stranded DNA. And this virus encodes a lot of proteins involved in DNA replication, including the polymerase, coded in the UL30 gene. The virus also encodes a single-stranded DNA binding protein, much like the one encoded by adenovirus, an origin binding protein, much like T antigen, a processivity protein, which helps make long DNAs, and 3, which are part of the primase to synthesize primers. And so all of these, with the exception of the origin binding protein, are not found in the smaller DNA genomes. This genome has 3 origins of replication. Now, we saw SV40 had 1, adenovirus had 2, 1 at each end. This virus has 3. It has 2 ORI-S and 1 ORI-L. This replication is unique because the DNA enters as a linear molecule, it's converted to a circle, and it replicates as a rolling circle. Let's take a look at how that works. So here again is the viral DNA on the top. It enters the nucleus. Remember, the virus uncoats at the cell surface, and the nucleocapsid is moved along microtubules to a nuclear pore. The DNA exits via the portal, and then the DNA is in the nucleus, where it's circularized by a cellular ligase called DNA ligase 4. So you go from a linear to a circular molecule. And it's thought then that this circle is the template for replication by rolling circle replication. And that works sort of like the name implies. You have a double-stranded circular DNA, and a nick is introduced, which gives you a free 3' end, and then that can be used to initiate as the primer for DNA synthesis, which is shown in red. As DNA synthesis proceeds, it displaces the top strand, And you can imagine this circle rolling as the new strand is made. This red strand eventually comes around and keeps going. Meanwhile, the 5' end can be copied by discontinuous DNA synthesis. And these two things occur at the same time. The red keeps going around and around the circle, and you get longer and longer 5' ends. And these are extended more than genome lengths. You have actually what are called concatemers. So that means there are multiple genome lengths. And you can imagine as this keeps rolling and you get more and more DNA made, you have more and more genome length entities. There's really no end problem here because all of these can simply continue to be extended. This is by RNA priming and the RNA removed and the gaps filled in and ligated and so forth. And we'll see later these are packaged into DNA particles, into viral particles by headfuls. This is threaded back into the particle by the portal, and then once you reach a genome length, it's cut. And we'll look at the mechanism for that later. Our last virus to talk about in terms of how the DNA is replicated are poxviruses. And I want to introduce them here because they're unusual in that they replicate in the cytoplasm. In poxviruses and many of the giant viruses, they do replicate in the cytoplasm only. So we'll see how that happens. And then the DNA is unusual. It has these terminal loops. So how does that replicate? Here's an experiment that shows you replication of poxvirus DNA in the cytosol, and what we call factories, because those sites of replication are not only where the DNA is made, but where particles are made as well. So on the left, we have an infected cell here in all three panels, a single infected cell. On the left, the cell is stained with DAPI, which stains DNA blue. And you can say that the nucleus is extensively stained with DNA, but there's also clearly some DNA in the cytosol. If this were not infected, you wouldn't see any DNA in the cytosol. And you can see these structures on the left there, they're clearly roundish structures that have DNA in them. And then the rest of the cell more diffuse. Then if you stain this same cell with an antibody to DNA binding protein, which is a virus-encoded protein, you can see there's nothing in the nucleus, at least none of that protein. But that protein appears to be in some of the same structures throughout the cytoplasm. Now, if you merge the blue and the red signals here, you can see that these areas in the cytosol have both DNA and DNA binding protein. So they're likely to be sites of virus DNA replication. And in fact, those are the DNA factories where virus particles eventually are going to be made as well. And so this virus does everything in the cytosol. It enters and sets up these factories in the cytoplasm. It has to encode a transcriptional machinery of its own, including RNA polymerase and transcription factors, so that it can make mRNA in the cytoplasm, and it makes DNA polymerase, everything you need for DNA replication. At least 15 viral proteins are involved in viral DNA synthesis for poxviruses. And the way we think this happens is beginning with a genome monomer. Primers are put down on the genome. These complete the second strand. Concatemer resolution occurs, probably via nicking and strand displacement, but we're not sure. But in the end, we have two monomers made. And again, the key here is that there's no end problem because these are circular DNA templates. Our next question is, what makes poxvirus DNA replication different from all of the other viruses we discussed today? A, the complete replication machinery is encoded by the viral genome. B, DNA synthesis occurs in the nucleus. C, DNA synthesis occurs by strand displacement. D, none of the above. The answer is A, the complete replication machinery is encoded by the viral genome. It's different from all the other viruses that we talked about today. The DNA synthesis, of course, occurs in the cytosol, and it's not by strand displacement. Let's take a closer look at origins of viral replication. We've looked at the replication of 4 different kinds of viral genomes, the polyomerases, Parvoviruses, Herpesviruses, Adenoviruses. In general, these origins are AT-rich parts of the genome. They're recognized by origin recognition proteins. The AT-rich is probably important because it's not as well base paired as GC areas, and so it can be more easily denatured so that you can copy the two strands. And as I've shown you, the origin is an assembly point for a DNA replication machine that is composed of many different proteins. As you saw with SV40, some virus genomes can have one origin, and we've talked about the herpesvirus genome, which has different origins. And you may wonder why the herpesvirus genome has two different kinds of origins. These work during different parts of the replication cycle. There are two distinct phases in herpes simplex viruses. There is the lytic replication, where you're making virus particles, and there's latent replication, where you don't make virus particles, and so they function during different times. Here are some close-up views of origins of replication. On the top, SV40, which we saw earlier, we have a core origin where we have an AT-rich region there in green. The large T binding sites, there are two different sets of that. And then on the left, part of the early promoter. In the middle, the herpes simplex L. Again, AT-rich sequences flanked by origin recognition protein binding sites. And in red, binding sites for transcriptional regulators. So you can see there are two promoters here flanking this origin of replication. In Adenovirus 5, the origin is at the end of the DNA. We have an area bound by origin recognition proteins, the polymerase TP. We have AT-rich sequences to facilitate denaturation. And here again, there's an early promoter around here, and these are binding sites for transcriptional regulators, immediate early promoter. The proteins, the viral origin recognition proteins we have talked about, polyomavirus T, large T. It's originally called T antigen, so sometimes I'll refer to it as that, or just large T. It binds specifically to DNA, so the origin binding protein in this figure is shown in red. It binds to DNA at the origin that recruits cellular replication proteins, as we've seen. The Parvovirus Rep68, 78 protein binds at the ends and unwinds the DNA. It's also involved in terminal resolution, releasing the product. There it is in red. Cellular replication proteins in green. Adenovirus pre-terminal protein binds at the terminus and actually recruits the DNA polymerase, which is also virus encoded. So pre-terminal there in yellow and the polymerase in purple. And finally, for herpesvirus, the UL9 protein recruits viral proteins to AT-rich origins and then unwinds DNA. So here we have the viral origin binding protein, and it's binding at this particular L. These viral origin recognition proteins have structural homology. Here are the three-dimensional structures of three viral origin binding proteins from AAV5. This, of course, is a parvovirus, from a bovine papillomavirus, which separate family from the polyomaviruses. SV40, large T, a member of the polyomavirus family. And you can see they look similar. The beta strands are colored in green, and you can see they have a core comprising a beta sheet, which is very similar, and around which there are similar alpha helices. And so these likely evolved from a common ancestor. The AAV5 origin binding protein is different in that it's also an endonuclease. Remember, it nicks the template and helps release the product. And so there's the endonuclease active site. So in addition to DNA binding, which is common among all three of these, the AAV5, the parvovirus protein, has an enzymatic activity. Let's take a close look at large T antigen of SV40. Here's a schematic of the protein, 708 amino acids long. This has been called one of the most studied proteins on Earth because so many people for so many years studied it and found many different activities and domains on the protein that regulate those activities. Now, we've talked about it today as a DNA binding or origin binding protein. Last time we talked about it as a transcriptional regulator, but it also turns out to be species specific. Pre-initiation complexes do not form in the wrong species. So if you have a monkey large T, it will not cause the formation of a DNA synthesis complex in a mouse cell. So that's what we mean by species specific. And the reason is that in the wrong cell, it doesn't interact with the primase. If you remember early on in the DNA synthesis scheme, the polymerase alpha is recruited in by large T, by interactions with large T, and that doesn't happen in the wrong species. Now, on the amino acid sequence of large T, the part of the protein that interacts with polymerase alpha is shown in tan. There's a piece at the N-terminus, and then there's a larger piece in the middle. So Pol alpha is the part of the region of this protein that interacts with that protein. And also you can see the origin binding sequence of this protein. There's a nuclear localization signal, of course, that gets the protein in the nucleus after it's made in the cytoplasm. There's the helicase domain where it helps, which is the part, of course, that denatures the DNA so it can be copied. And that activity requires ATP, so there's an ATPase domain as well. But another very interesting aspect of this protein is that it binds and sequesters proteins that regulate the cell cycle. And that causes cells to enter S phase. The part of this protein that binds the cell cycle regulators is right here. It's in tan. It's labeled RB. How that works, we're going to see in a moment. There's also a place for binding P53, which we'll talk about when we discuss transformation. But for now, how does T antigen binding to RB cause cells to enter S phase? And why would it do that in the first place? Well, first of all, you have to understand that most of the cells in our body are quiescent. They don't divide, and when they do, they don't divide very often. Of course, our neurons don't divide at all. Many of the cells in our muscles and other organs don't divide. Exceptions are, for example, epithelial cells that line our mucosal areas, like our gut tract. Those cells divide frequently. But most cells are not dividing. In turn, viruses don't replicate well in cells that are not dividing. Remember, most DNA viruses need at least something from the host cell, some protein. The smaller ones need way more than the others. If a cell is not dividing, it's not making replication proteins. So smaller viruses induce host replication proteins. By smaller, I mean viruses with smaller genomes. And they get the cells dividing. This is done by early gene products like T antigen. Getting the cell to divide, I call it kicking the cell into cell division, is done by early gene products like SV40 T antigen. So here is a diagram of the cell cycle. As you know, cells divide by mitosis. Starting from a single cell, the chromosomes duplicate, they're separated, and then the cells divide to make two from one. The cell cycle takes about 24 hours and is divided into distinct phases. Called M here for mitosis, where the actual division occurs. But then that's followed by G1, a period of cell growth, an S period where DNA is replicated, and then a G2 just before mitosis. This cycle is controlled by a complex series of cell proteins because cells don't need to divide all the time, so you have to regulate mitosis. And a key regulator is a protein called RB, standing for retinoblastoma. There's a restriction point right here in G1, which is governed by the RB protein, which will not allow the cycle to proceed into S unless certain conditions are met. And some of those conditions include that there are enough nutrients in the surrounding the cell to permit proper cell growth. This protein was originally identified in children with tumors, eye tumors, called retinoblastomas. And its loss was associated with the formation of tumors. So kids who developed this particular kind of tumor had loss of function mutations in this gene. We call it a tumor suppressor gene. Its presence is needed to suppress tumors because it's needed to control cell growth. We'll talk more about that later when we discuss transformation. So what does that have to do with viruses? If you remember from when we talked about transcription, the adenovirus E1A protein binds RB to liberate E2F, which it needs for its early gene transcription. Now, E2F in the cell is typically complexed with another protein called DP, but it's more importantly complex with RB protein. When this E2F binds cell promoters, RB recruits histone deacetylases and represses transcription from cell promoters. And these promoters are typically in genes that control the division of the cell, including cell cycle specific kinases and many others. So this promoter that we're showing here is driving the synthesis of mRNAs for genes, including cellular replication proteins needed for DNA synthesis and passage through the cell cycle. For adenovirus, the promoter also requires E2F, and hence it would be repressed by RB, and that's why the virus produces E1A to get RB off of E2F, so now early gene transcription can occur. The cell does the same thing when it needs to divide. RB is phosphorylated. That knocks it off of E2F, and now cell promoters that encode replication or mitosis proteins can now be transcribed, can now be active. Similarly, large T binds RB, not because it needs E2F, as adenovirus does. SV40 large T binds RB because that will now turn on transcription of cellular genes that help pass the cell through the cell cycle, including DNA synthesis genes, DNA polymerases and auxiliary proteins that are needed for viral DNA replication. So in a quiescent cell where DNA synthesis is not occurring, the virus comes in, makes large T, and that turns on these promoters for DNA synthesis and makes the DNA synthesis genes that the virus requires for its own DNA synthesis. So that's another function of large T. In addition to binding the origin, denaturing the DNA, being a transcriptional promoter, it is also binding RB and turning on genes in the cell that encode DNA synthesis proteins. And in fact, adeno E1A does the same thing. Besides liberating E2F for its own promoter, it also turns on DNA synthesis genes. And the papillomavirus E7 protein similarly binds RB to turn on DNA synthesis in the cell. For the smaller viruses that don't encode many proteins needed for DNA synthesis, they have to encode a protein like large T, like E7, like E1A, that binds RB, bypasses that checkpoint in the cell cycle, turns on genes that are needed for DNA synthesis in the cell and cell division, and those proteins are then used for viral DNA replication. This is quite an interesting activity, and as we'll see when we talk about transformation, it can accidentally lead to cells that have oncogenic potential. Next time, we're going to talk about the class of viruses that encode reverse transcriptase that copies RNA genomes into DNA and integrates them into the host cell genome. Reverse transcription and integration.